Good morning. I'm going to go over how to calculate the size of a cheek cell. Field diameter. Let's start with this slide. All right, field diameter is the circles that we see in this image. Those represent what you would be able to see under the microscope, the area within the circle. So the red circle represents what you're likely to see. So between red circle here and the other end of the red circle, the diameter. That's what you would be able to see if you were viewing a specimen under the microscope at 10x of field diameter. So we could see a comparison at 100x total magnification at 10x objective versus how much you could see at 40x objective. So the difference here, you could see that here you see a lot more of the area. You see a lot more cells because you see a lot more of the slide. When you magnify it to 400 times magnification, or we're at the 40x objective, we see less cells. And so that's the black circle. And the line that I just drew is the diameter of the 40x objective, or the 400 times magnified field. So this slide just shows you how much you could see under the microscope according to which objective you're using. We go to this field that shows you measurements. Normally in lab, we would literally put a little tiny ruler on a slide, look it under the microscope, and measure that field, that circle that you see under the microscope. We're not in lab, so we gave you that image here with labels. So we look at the big circle right here, we see that that's a scanning power field. So those are objects that are magnified 40 times. We're in the 4x objective. Things are magnified a total of 40 times. And we see that that has a measurement. So from this line here to this line here, that's 1 millimeter. So we could calculate the field at this magnification. So 1 millimeter, 2 millimeters, here to here, from here to here is 3, here to here is 4, here to here is 5. So the total the field diameter for 40x, the scanning power is 5 millimeters. Then we could do that, so we'll put 5 millimeters there. We could do that for low power. The low power lens is the 10x objective. And when we magnified the image, it's 100 times magnified. And so that's the green circle here. Now, within this image, we could estimate the size. We could estimate the size. So I'm going to say I know it's for sure from here to here is 1 millimeter, and then I just have to calculate this area and this area. So let's just say 1.5 millimeters. And these are estimates. And then we go on and we look at the high power. So the high power is this yellow circle. The high power, the yellow circle. So the yellow circle is less than 1 millimeter. And so I'm going to estimate that's a 0.75 millimeters, 3/4 of a millimeter, 0.75 millimeters. And lastly, we're going to look at the 100x objective. Things are magnified 1,000 times in this view. And I'm just going to estimate that that's 0.4 millimeters. The rough estimates. Which one's the one that I do know for sure? The only one that I really could say for sure is the scanning power, right? That's the 5 millimeters. And then this one's an estimate too. That's not too off. So this is what we need to measure the size of a cheek cell. We need a ruler, and this is where we get the ruler, and this is where we do the measuring. So we could measure objects under the microscope for example, that cheek cell. So we'll go through this total magnification versus diameter depth. This explains that when you're on objective or objective, which is the scanning objective, your total magnification is 40 times. You've magnified that image 40 times. That's the widest. We just saw that previously. And then it gives you information on the depth of field. Likewise for each of the subsequent 10x, which is the low power lens, magnifies images 100 times, and then the size difference decreased by a factor of 40 over 100. All right, so field diameter and pointer. Okay, the field diameter is what we calculated in the previous slide. The pointer is half the radius. So under the microscope, you have your field, and usually you have a pointer. And the pointer, it looks like a line that takes up half the space of your field. That's the radius of the diameter, and the radius of a diameter is half the diameter. So check. In this slide, it refers you to the slide that we calculated the measurements, the 5 millimeters, 1.5 millimeters. Okay. So what is the power length on low power? I'm sorry, what is the pointer length on low power? Let's go back. Now that we've calculated the diameter, so the distance between one end of the field and the other one, for 40x, it's 5 millimeters, 100x is 1.5 millimeters, 400x is 0.75 millimeters, and for all immersion length, it's 0.4 millimeters. Again, those are rough estimates. Usually, when we're looking under the microscope, the image, the field has a pointer. And so that pointer is the radius of the diameter. And the radius of the diameter is half the diameter. So whatever the diameter is, divide by 2. And that's the radius of the diameter. So for 40x, the radius is 2.5 millimeters. For 100x is 0.75, and etc. So that's the radius. That's how to calculate the radius. Now we go on. Okay, so let's go back to the field diameter of 400x is what we need to calculate the size of the cheek cell. Okay, and the way you calculate that is the diameter of the high power equals diameter of low power, which we'll get from the previous calculations, times the power, how much it's magnified, of low power lens. The low power lens is the 100x objective, at the 10x objective, 100x magnified, divided by the power of the high power lens, 400. And that's how you're going to get the diameter of the high power. So what is the pointer length of 400x? Okay, the point of the 400x, that's the high power lens. That's what we're calculating, which we did earlier. The pointer length is the radius, so the pointer x is at 100x, 0.75 divided by 2, so 0.375. So that's your answer for this one, 0.375 millimeters. Okay, so that was field diameter and pointer length. Right, so recall that we calculated already the diameter of each of the powers, 40x magnification, 100x magnification, 400, etc. We calculate the radius of each, the pointer length, by just dividing by 2. Now here's an equation for diameter of high power. The diameter of high power, the 540x, remember that that's the yellow area here. And we estimated, we know it's less than 1 millimeter according to this image, but we estimated. So a little bit more accurate calculation is we follow this equation. Diameter of high power, so that's our unknown, what we're looking for, equals diameter of low power. The low power is 100x, so diameter of low power is 1.5 times the power of the low power and divided by the power of high power lens. So we calculate here the high power diameter, 1.5 times 0.25, and that's high power diameter, so diameter here, equals 0.375, which matches pretty closely to our rough estimate, but now we have a calculation for it. So we know the diameter of the area in yellow, the high power field. And we need to know this in order to calculate the size of one individual cheek cell. Okay, So we used here the diameter of low power, and our answer is diameter of high power equals 0.375 millimeters, which is very close to what we estimated. Measuring the size of cheek cells. In order to view a cheek cell, and these are your epithelial cheek cells, so we take a scraping from the inside of your cheek, mix it with methylene blue dye, and these are live cells. They take up the dye, and this is the image you see of the cells. They're eukaryotic animal cells, so they fold up when you spread them on a slide. You put a little cover slip on top, and you view them under the microscope. So this is normally what you would see. Here it would be too clumped in order for us to really see it, so we look for an area where we see them a little bit more spread out. All right, so we want to calculate what is the size of one epithelial cheek cell. Here's this image that you would see under magnified cells are magnified 400x. So we're in the 40x objective. Okay, so magnified 400x. And your field, what you would see under the microscope, is outlined by this circle in black. Okay, so how many cheek cells can you fit along the pointer? The pointer is right here. All right, if this image was taken at 400x total magnification, what would be the field diameter? How long would the pointer be? Now, how long is the field diameter? We calculated the diameter, right? Before, high power diameter equals 0.375 millimeters. How long would the pointer be? The pointer would be half of that, so 0.1875. Now divide the pointer length by the number of cells that fit in the pointer. So here I'd see, so here divide, how long would the pointer be? How long would the pointer be? Let me write that down, 0.1875. And how many cells would fit under the pointer length? I would say 1.5. So divide that by 1.5. And you get the size of one cheek cell equals 0.125 millimeters. Calculating the size of the cheek cell. So let's go over it. We know the diameter of the high power lens. We calculated that previously. And so calculate the size of the cheek cell. The size of the cheek cell equals field diameter of the field that you're looking at, in this case at a 400x, divided by number of cells that fit across the diameter. So this differs from this method, but you get the same answer. Okay, so what is the size of the field? Size of cell, one cell, equals size of field. We said that the size of the field is 0.75, calculated that earlier, divide by number of cells that fit across the field. Now I see that one fits here, and this one, if I moved it a little bit, it'd take up about that much space. So I think I could fit 2.5 cells here. So I'm going to divide by 2.5 cells. So 0.375 divided by 2.5 equals millimeters for one cell. So that's the size of one cell. Now, in order to convert this to micrometers, then we need to know how many micrometers fit in 1 millimeters. That's 1,000 micro. So you multiply by 1,000 micrometers. And that size of one cell is 150 micrometers. So this is how you calculate your cell.